All life is made of one or more cells. This amoeba pictured carries out all of its life functions within a single cell. While other organisms like us are built with a variety of cell types, the total number of cells numbering in the trillions. Just like a Lego toy set is made of individual Lego pieces of various types, we are made up of cells of various types. Though cells are diverse in appearance and function, they all have the same basic structure. This chapter will delve into the inner workings of the cell, differences between prokaryotic and eukaryotic cells, as well as plant and animal cells. Cells are too small to be seen by the naked eye. We need to use microscopes in order to magnify cells. Because of the optics of light traveling through air and glass, the image may appear to be in a correct orientation on a slide, but it appears upside down to us. This has to do with the design of the microscope correcting the pathway of light. If you were to move a slide to the right, it would appear as if it was moving left under these microscopes. If you take any one of my classes at Maysville Community and Technical College, you'll be using a Nikon E200 microscope, as pictured here on the right. A little bit more about the terminology of microscopy. The obvious thing microscopes do is it brings us closer to smaller objects by magnification. Magnification is how close you are to an object compared to the naked eye. So the naked eye sees things in 1x. I'm going to start off with something simple, such as a photo. Here's a picture of a tiger that I took at the Columbus Zoo many years ago. You'll notice the field of view, which in microscopy is the area under the microscope, but in this photo is the exhibit itself. You'll notice the field of view is pretty large, but the tiger itself is smaller. But if I were to zoom in and magnify that tiger, the field of view gets smaller. And that's a relationship that we see between the field of view and magnification. As the field of view decreases, your magnification increases. Here's some red blood cells at 400x, that is 400 times closer than the naked eye. And here they are again at 1000x. Now think about these two pictures, is even though there's more red blood cells in the left photo, they're smaller, and there's fewer red blood cells in the right photo, but they're much larger in size. The next concept is resolution, and that's the degree of clarity of an object. Notice how the use of a light microscope versus an electron microscope, which I'll talk more about next, differs in clarity, even though the same object is viewed under the same magnification. In most biology labs and clinical settings, a compound light microscope is used to view cells. In my biology lab, our microscopes can see up to 1,000 times. We can even view colonies of bacteria. Pretty cool. The advantages of light microscopes is that they are cheaper and can view live organisms. The disadvantage of light microscopes is that they're usually limited in magnification, and if an organism is hard to see, it will have to be stained, which ultimately kills the organism. Electron microscopes use a beam of electrons instead of light. The advantages of an electron microscope is that you can magnify an object much further than using a light microscope. It also has a higher resolution. The disadvantage of an electron microscope is that they're really expensive and can't be used to view living materials. So cell theory is one of the major pillars of biology, and it's been around for quite a while. But it all started off with this man named Anthony van Leeuwenhoek. In the 1600s, this man, a curtain and drapery merchant, used a particular tool that he invented to help understand the fine threadings of the drapes and curtains that he was working with. He was also an amateur scientist, and at the time he was dealing with a bout of diarrhea and was curious, so he examined his own stool and found this particular organism right here. Now, bear in mind that this would not have been what he had seen based on that tool. This is an updated version that you would see using our microscopes. He called these animalcules, and later discovered things like sperm cells, bacteria, and the banding patterns on muscle cells. So if we look at this organism, it's Giardia lamblia. He most likely had the disease giardiasis, which is a waterborne disease caused by contaminated water. While Leeuwenhoek is credited for seeing the first microscopic organisms alive, Robert Hooke used his microscope and first discovered microscopic life by taking a sample of plant tissue. He observed the pattern under the microscope of what he described as honeycombs and called them cells, since they resemble gel cells. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann studied the tissues of several plants and animals and developed what we still use today as cell theory. The main components of cell theory are: all organisms are composed of cells. Cells are the basic units of structure and function in organisms. In other words, what happens at the organism level happens at the cell level. What happens at the organ level happens at the cell level. Finally, cells come only from pre-existing cells because cells are self-reproducing. What that means is that human cells can only make human cells, dog cells can only make dog cells, muscle cells make muscle cells, skin cells make skin cells. But we have to be careful because all cells, all life starts off as stem cells. Stem cells are undifferentiated cells, meaning they do not have any particular job. And so therefore, once they do receive that particular job, they become differentiated. And once differentiated, they can only make cells like themselves. Cells can be broken down into two major groups. But before we get into that, it's important to understand what all cells have in common. First, a plasma membrane, which is a barrier to help separate the interior from the exterior of the cell. A cytoplasm, which makes up the fluid inside of the cell. DNA, which holds the genetic information of the organism, and ribosomes, which takes information from DNA to make proteins. Cells fall into two major categories: prokaryotes or eukaryotic cells. Prokaryotic cells make up single-celled organisms like bacteria and archaea. Eukaryotic cells make up organisms like plants, animals, fungi, and protists. This table displays the major differences between both groups. You'll see that organelles, or membrane-bound compartments, are absent in prokaryotes, while eukaryotes contain several. In prokaryotic DNA, it is circular and found in the region called the nucleoid, which is not readily defined. In eukaryotes, DNA is linear and located in a membrane-bound nucleus. With very few exceptions, prokaryotes are much smaller than eukaryotes, and to date, all are single-celled. Eukaryotic cells can be on average 10 times bigger than prokaryotic cells, and while single-celled eukaryotes exist, most eukaryotic organisms are multiple-celled. So let's focus on prokaryotic cells. These were most likely the first types of cells found on planet Earth and have been around for 4 billion years. First, I want to direct your attention to the nucleoid. In this region, it holds the genetic material. And again, nucleoid means nucleus-like. It's not actually a defined structure. It's just an area where there's some free-floating chromosomes. In addition to the nucleoid of prokaryotes is a plasmid, and this is only found in prokaryotes. In addition to the nucleoid, prokaryotes can have plasmids, which are separate strands of circular DNA, and these are exclusive to prokaryotes. Outside of the plasma membrane are a couple of layers of protection. First is the cell wall, which also provides the shape to the prokaryotic cell, and many prokaryotes have a protective capsule, which protects prokaryotes from dehydration, as well as providing more reinforcement to the prokaryote shape. This is made of a special carbohydrate called peptidoglycan. Flagella are used for locomotion, while pili are used to help transfer genetic information between prokaryotes in a process called conjugation. While prokaryotic cells only reproduce by asexual means, the ability for them to transfer genetic material is all owed to the pilus. So, prokaryotic cells are much smaller in size than eukaryotic cells. While most cells are too small to be seen without a microscope, it's understandable to ask, why are cells so small? Well, it has to do with surface-to-volume ratio. As a cell increases in size, its surface-to-volume ratio decreases in size. Cells bring in materials and release waste by diffusion. The smaller your surface-to-volume ratio, the more efficient it is to do these processes. The only way for a cell to survive is to divide when it gets too large. And it's important to know, larger organisms do not have larger cells, they just have more of them. In our next video, we're going to look at the specifics of eukaryotic cells.